Hi students. In today's class, we will learn about the structure and types of nephrons. By the end of this class, you will understand what are the different parts of a nephron and what is the importance or relevance of these different parts with regard to the function which they perform. Okay. Nephron is a functional and a structural unit of a kidney. In the last class, I told you that each kidney has about 1.5 millions of nephrons. If you take each single nephron and study the structure of a nephron, you will see certain parts. And we will, in today's class, understand in detail about each part. Before that, let me tell you a small analogy. Nephron is basically working like a filter. Okay. Now, I want to compare this filter action with the usual filter what we use in our kitchen. But in fact, both are uncomparable. Let me tell you how. Say you take a filter which you use in the kitchen to filter out the tea. What will you ultimately do? You put the milk through the filter and discard the things which are above and consume the tea. Am I right? Similar to this, each nephron has got a filter through which blood is now filtered. Okay. And what are we going to do? The fluid which is collected beneath the filter, the kidney will take out the required material, eliminate the unwanted or waste material, and the required material whatever is taken is added back to the blood which was above the filter. Okay. These processes technically are named like this. I wish you make a note of it. The first step where blood is filtered in a nephron, the upper part of the or the superior most part of a nephron is called glomerular filtration. That means the blood enters and there is certain filtration which has occurred. Now, what is remaining here? The bigger masses in the blood like blood cells and proteins. Now, the part which is filtered below this filter or filtration membrane, this filtrate is now called ultrafiltrate. Okay, from this, certain parts should be taken back, right? Because they are important also. So the parts which are taken back and added to the blood is called reabsorption. So you are absorbing them again, reabsorption. Okay, and certain parts of the nephron, that means the distal part, okay, wherein the toxic materials are taken and to be added, this is called secretion. So after filtration and reabsorbing the required material, whatever is left out is eliminated in the form of urine. Okay, this is what nephron is doing. So keeping this basic idea, I know it will be vague in your mind right now. Just keep this idea and then follow me through a diagram. I will try to explain to you where and how each of these functions are happening. Before that, recollect from the previous discussion that the nephrons are basically of two types. What are they? Cortical nephrons, you remember, 80 to 85%, and medullary nephrons, more precisely called juxtamedullary nephrons, which are constituting about 10 to 15% in each kidney. The cortical and the juxtamedullary nephrons are quite different in the way they are. Why? The functions of them are also quite different. Okay. So when I explain to you the structure of a nephron basic outline idea, I will also tell you where is the cortical nephron and medullary nephron are differing in their structure. Okay. So without further delay, let us understand about the structure of nephron. In this illustration, I am going to explain to you about the structure in detail, that is the microscopic or histological structure of nephron. I will try to deal with the structure of nephron based on its function. So, by the end of this illustration, you will be able to appreciate that the histological differentiation of different parts of nephron is actually to do different functions. Recollect quickly that the kidney has a renal cortex and a renal medulla. The medulla is formed into renal pyramids. and renal cortex will intervene in between as renal columns. The nephrons are arranged both in the cortex as well as in the medulla. 85% of the nephrons are cortical nephrons who have the short loops of Henle and but the collecting ducts are longer. Whereas 15% of the nephrons are juxtamedullary nephrons like what you are seeing this in the yellow figure. It has a very long loop of Henle extending up to the tip of the papilla and again a very long collecting duct. So, when 85% of the cortical nephrons are doing the basic functions of glomerular filtration, tubular reabsorption and secretion, 15% of the juxtamedullary nephrons, as we shall see later, are exclusively involved in countercurrent mechanism. Alright, each nephron will receive a afferent arteriole which gets into the first part of the nephron called renal capsule or Bowman's capsule. Bowman's capsule. Students also recollect that the afferent arterioles are the branches of interlobular arteries. As the afferent arteriole gets into the Bowman's capsule or the renal corpuscle, it splits up into multiple capillaries. The capillaries are arranged in the form of a spear, usually called as tuft of capillaries. They are arranged like a ball or a sphere, tuft, which fits into the concavity of the Bowman's capsule. Usually what we know that an arteriole in the circulation in general will give rise to capillaries. They fuse to form a venule. But students, here only in case of kidney that an arteriole after giving rise to capillaries will give rise to another arteriole, efferent arteriole in this case. As you can see, afferent arteriole gets into the Bowman's capsule, forms glomerulus and leaves the glomerulus, leaves the Bowman's capsule in efferent arteriole. Okay, this is the first peculiarity you must know. Now coming to the structure of the nephron. We will first see what is this Bowman's capsule. The Bowman's capsule has got two layers, an outer parietal layer and an inner visceral layer. So this is a visceral layer, outer is parietal layer. What do I mean by visceral layer? That layer which is in close contact with the capillaries. Parietal layer is outside. Between the parietal and the visceral layers, there is a space called Bowman's space. Into this space that the plasma will get filtered and is called ultrafiltration. Okay. There are certain special modified epithelial cells in the visceral part of the Bowman's capsule or the visceral layer of the Bowman's capsule. They are called podocytes. As the name suggests, you can see this podocyte cell has got foot processes which go away from the cell body, and these foot processes will further form flat processes which are interdigitating. That means the foot processes of one podocyte and the foot processes of the other podocyte will interdigitate, something like crossing your fingers, and there will be small narrow gaps or pores through which the filtration occurs. After the initial part, that is Bowman's capsule, the next part of nephron is called proximal convoluted tubule, the one which is shown in the yellow color in this picture. The proximal convoluted tubule is convoluted, as the name suggests. It is not a straight tubule, it is convoluted. But in the last part of 1/3, it is a straight segment. So the first 2/3 of it is convoluted, but the last part is in fact a straight segment. The cells in the proximal convoluted tubule, as you can see here, are closely placed and they are having tight junctions between them, which are selectively permeable or leaky only to certain electrolytes. The cells of the proximal convoluted tubule also have a very high concentration of mitochondria in their basolateral side, which keeps giving energy in the form of ATP to operate sodium potassium ATPase pumps. The surface of the proximal convoluted tubular cuboidal cells is thrown into multiple villi to increase the surface area for reabsorption. This is called the brush border of the proximal convoluted tubular cells. That is the reason why proximal convoluted tubules, because they are having a very huge amount of brush border and mitochondria, they are able to do about 60 to 70% of total reabsorption in the nephron. The proximal convoluted tubule will lead to another structure called loop of Henle, the one which is shown in the green here. The loop of Henle has got a descending limb, a hairpin bend, and an ascending limb. If you can see, the ascending limb is further divided into a thin part and a thick part. A thin ascending limb and a thick ascending limb. As I told, the function of loop of Henle is basically to create an osmolality gradient and involve in a mechanism called counter current multiplier system to produce a concentrated urine, which we shall learn in the later classes. Coming to the histology of the cells in the descending limb of loop of Henle, the hairpin bend and the thin ascending part, they are all flat cuboidal type of cells with a few brush border or very few villi. Their main task is to produce a concentrated urine. The thin descending limb of loop of Henle, which we are seeing here, is highly permeable to water, whereas the thick ascending limb of loop of Henle is impermeable to water because the cells here are again cuboidal and having very tight junctions. The loop of Henle will lead to another convoluted part called distal convoluted tubule. So this is proximal convoluted tubule shown in the yellow and this is distal convoluted tubule shown in the orange color. The distal convoluted tubule is having again cuboidal cells which are having very less amount of brush border and only few mitochondria. They, like the proximal convoluted tubule, are also involved in the reabsorption of important solutes like sodium and chloride. Sometimes what happens? When the ascending limb of loop of Henle is coming into the cortex, that means from the medulla towards the cortex, when it is ascending up, what happens is the distal convoluted tubule will come in close contact with the afferent arteriole or the efferent arteriole or both. When such a thing happens, a close contact between these two will exist, then the cells in the DCT will get modified. The cuboidal cells will get modified into low or tall columnar cells which are very closely packed. Such modification is called macula densa. Macula densa. These cells will be sensory in nature. They keep sensing the sodium amount in the distal convoluted tubule and they take part in a system called juxtaglomerular apparatus, which we shall learn in the later classes. Many distal convoluted tubules will open into a single collecting duct, the one which is shown in the blue color. The collecting duct cells will not have the brush border, they are simple cuboidal or squamous type of epithelium. The collecting duct can also be further divided into a cortical collecting duct close to the PCT, DCT or the Bowman's capsule and a medullary collecting duct which goes deep into the interstitium up to the renal papilla. The cortical collecting duct cells which are basically two types, the principal cells and intercalated cells, are doing two important functions. The principal cells are the target for antidiuretic hormone and they help in reabsorption of water, whereas intercalated cells are involved in acid-base balance. Students, remember that many distal convoluted tubules are opening into few collecting ducts. That means both the kidneys have together 1.5 millions of nephrons. That means each nephron has will end in the distal convoluted tubule, but it has few millions or less than 1 million or thousands of collecting ducts because many distal convoluted tubules are opening into few collecting ducts. And collecting ducts, what do they do later on? Multiple collecting ducts will open into a common duct called papillary duct, which is at the tip of the renal papilla.